Imagine holding in your hands a fragment of parchment so old that the civilization that produced it had already crumbled to dust more than 1,000 years before your grandparents were born. Imagine that this fragment contains words, ancient words, words that scholars have spent entire lifetimes trying to read, to place, to understand. Now imagine that hidden inside the very skin of that parchment, invisible to the naked eye, invisible even to the most powerful microscopes of a previous generation, there lies a second story, a biological story, a story written not in ink but in DNA, a story that no scribe ever intended to leave behind, and yet one that has been waiting patiently, silently, for 2,000 years, for someone with the tools to finally read it. That moment has now arrived, and what scientists found when they read it shook the foundations of everything we thought we knew about one of the greatest archaeological discoveries in human history. The Dead Sea Scrolls are not simply old documents. They are the oldest known manuscripts of the Hebrew Bible ever discovered. They are the textual bedrock upon which centuries of religious tradition, scholarly interpretation, and cultural identity have been built. When they were first unearthed beginning in 1947, the world held its breath. Bedouin shepherds stumbled upon clay jars tucked inside the caves of Qumran, along the northwestern shore of the Dead Sea, in what was then British Mandatory Palestine. Inside those jars were scrolls of leather and papyrus, wrapped and preserved by the extraordinary dryness of the Judean desert. What they contained rewrote the timeline of the Bible itself. Texts from the book of Isaiah, from Psalms, from Deuteronomy, some dating back more than 2,100 years, were suddenly in human hands for the first time since they had been hidden away. The scholarly world erupted. Religious communities trembled with anticipation and anxiety in equal measure. For decades after that initial discovery, archaeologists and biblical scholars worked to piece together what they had found. More than 900 manuscripts were eventually identified across 11 different caves, the fragments numbered in the tens of thousands. Some were large enough to display coherent columns of ancient script. Others were no bigger than a fingernail, blackened and brittle, defying every attempt at reconstruction. The conventional understanding that emerged from this monumental effort was powerful but incomplete. Scholars believed that the scrolls had been produced by a single Jewish sect, most likely the Essenes, a monastic community that had retreated from Jerusalem society to live in austere devotion at Qumran. The story was tidy, it was coherent, it had for a long time the comfortable ring of consensus. But beneath that consensus, questions had always festered. Why, for instance, were some of the scrolls written on leather that appeared to come from different animals entirely? Why did certain texts seem stylistically and theologically incompatible with one another, as though they had been composed by communities with sharply divergent beliefs? Why did the handwriting shift so dramatically across certain manuscripts? Traditional paleography, the science of dating and identifying texts through the analysis of handwriting and script style, could tell scholars something about when a document was written. It could tell them something about how, but it could not tell them where the materials came from. It could not tell them whether a scroll produced in Jerusalem had somehow been transported to Qumran, or whether a scroll found in one cave had originally been stored in another. It could not answer the most fundamental question of all: were all of these scrolls really the product of a single isolated community, or were they something far more complicated, far more extraordinary, far more disruptive to the established story? For decades, those questions had no answer. The technology simply did not exist to pursue them. Scholars debated, theorized, and published, but the scrolls themselves held their secrets with perfect geological patience. Then in the second decade of the 21st century, something changed. Not in the caves of Qumran, not in the archives where the scroll fragments were stored under climate-controlled conditions, but in the laboratories of molecular biologists. Scientists had been refining the techniques of ancient DNA analysis to a degree that previous generations would have considered science fiction. They had successfully extracted genetic material from Egyptian mummies, from Neanderthal bones, from the preserved remains of woolly mammoths, locked in Arctic permafrost for 40,000 years. The question that a small and bold group of researchers finally dared to ask was this: could those same techniques be applied to the Dead Sea Scrolls? Could the parchment itself, the animal skin upon which those ancient scribes had pressed their ink, yield DNA that had survived 2 millennia of desiccation, oxidation, and fragmentary decay? The answer, it turned out, was yes, and nothing would ever be the same. The process required extraordinary delicacy. The scroll fragments are among the most precious and irreplaceable objects on Earth. Any sampling had to be performed with near-surgical precision, extracting the absolute minimum of material, while causing no visible damage to the document. Scientists took tiny samples. Kine samples, in some cases less than a single square centimeter, from the edges or damaged zones of dozens of different fragments. They then subjected these samples to a process called next-generation sequencing, a technology so powerful that it can reconstruct genetic information from molecules that have been broken, degraded, and chemically altered over centuries. From these degraded fragments of biological material, they began to read something astonishing. They began to rid the genome of the animals whose skins had become the pages of history's most sacred library. What they found in the first wave of results was already remarkable. The vast majority of the scrolls had been made from sheepskin, but not all of them. Some were made from cowhide, which was unusual because cattle were not commonly raised in the arid environment around Qumran. This was the first crack in the wall of the single community theory. If the Essenes at Qumran had produced all of these texts themselves, using locally available materials, why were some of the scrolls made from the hides of animals that would have had to be sourced from somewhere else entirely? The DNA was pointing towards something that handwriting analysis and radiocarbon dating had never been able to suggest, that some of these scrolls had not been made at Qumran at all. They had been brought there. They had traveled. They were refugees of a larger, more dispersed literary culture, gathered into the caves not as the output of a single sect, but as a library, a collection, perhaps even an archive assembled in desperate haste before some catastrophe swallowed the world those scribes had known. But the story grew stranger still, and more profound, when the researchers turned their attention to the Great Isaiah Scroll, one of the most famous and best preserved of all the Dead Sea Scrolls. They made a discovery that stopped them cold. The Isaiah Scroll is a single continuous document, 54 columns of beautifully preserved Hebrew text, containing the complete book of Isaiah. It had always been treated as a unified manuscript, the work of one scribal tradition, perhaps even one scribal hand. But when the DNA analysis came back, it revealed something that no eye had ever detected. The first half of the scroll and the second half had been made from the hides of different animals. Not just different individual animals, but animals that were genetically distinct enough to suggest they had come from different herds, different locations, possibly different regions entirely. The scroll that the world had long treated as a single unified document was, at the molecular level, a seam, a joining of two separate pieces of parchment that had begun their existence far apart from one another and had been stitched together into the appearance of seamlessness by hands that knew exactly what they were doing. This discovery did not diminish the Isaiah Scroll. It transformed it, because now scholars began to ask a question they had never had the tools to ask before. If the parchment came from two different places, did the text? Were the scribes who wrote the first half of that document the same people who completed the second? And if not, what does that tell us about how the Hebrew Bible was assembled, transmitted, and canonized in the centuries before the Common Era? These were not merely academic questions. These were questions that reached into the heart of how billions of people around the world understand the origins of their sacred texts. The DNA was not just identifying animals. It was mapping the hidden geography of a civilization's intellectual and spiritual life. As the analysis continued, researchers began to find patterns that grew increasingly difficult to explain within the old framework. Certain scroll fragments that had been stored in different caves, and therefore assumed by many scholars to have belonged to different communities or collections, turned out to share remarkably similar genetic profiles in their parchment. This suggested that those fragments had once been part of the same document, the same library, perhaps even the same room, before some unknown force had scattered them across the Judean landscape. The caves of Qumran had not been separate depositories maintained by separate groups. They appeared, in the genetic record at least, to be the fragmented remnants of something that had once been whole. Something that had been deliberately dispersed, hidden in multiple locations, as though someone had known that the world they lived in was about to end, and had done everything in their power to ensure that what they valued most might survive it. That world did end. In the year 70 of the Common Era, Roman legions under the general Titus crushed the Jewish revolt, destroyed the temple in Jerusalem, and systematically dismantled the communities and institutions of Judean religious life. It is in this context that the scrolls take on their most heartbreaking dimension. Whoever hid those manuscripts in those caves was almost certainly trying to save them from the fire. And what the DNA analysis now suggests is that the people doing the hiding were not a small, isolated sect acting alone. They were part of a broader network, a wider world of Jewish scholarship and literacy, drawing text from multiple traditions, multiple regions, multiple herds of animals across the ancient Near East, binding them together into a record of everything they knew, everything they believed, everything they refused to let the Romans take from them. The scrolls are not the output of a monastery. They are the last transmission of a civilization standing at the edge of its own destruction. This realization carries implications that extend far beyond the archaeology of 1st century Judea. Because if the Dead Sea Scrolls represent a broader cross-section of Jewish literary culture than previously understood, then the texts they contain are not the exclusive product of one radical sect's particular theology. They are a window into the full, complex, contested, and deeply plural world of Second Temple Judaism, the world in which the foundations of both modern Judaism and early Christianity were being laid simultaneously, in competition, in conversation, in conflict with one another. The DNA analysis does not simply tell us where the animals came from. It tells us that the people who collected these texts were engaged in something far larger than the production of a sectarian library. They were trying to preserve an entire intellectual inheritance. And then there is the question that the science itself cannot answer, but that it forces us to confront. When those scribes stitched together 2 pieces of parchment from distant herds to create the Isaiah scroll, were they aware of what they were doing? Did they know that the material they worked with was composite, assembled from the scattered resources of a fractured world? Or did they simply do what writers and teachers and priests have always done? They took what was available, they used what they had, and they tried to pass something true on to whoever came after them. That question will never have a definitive answer, but it is a question that DNA has now made it possible to ask, and the asking of it changes us, because that is what this discovery ultimately reveals. Not just something about the Dead Sea Scrolls, not just something about ancient Judaism or the origins of the Bible, but something about the nature of knowledge itself. We have always known, in the abstract, that history is incomplete, that the record we have of the past is partial, accidental, shaped as much by what survived as by what was true. But there is something uniquely powerful about watching a piece of parchment, a dead piece of animal skin 2,000 years old, yield up a biological secret that redraws the map of an entire civilization. It tells us that the past is never as finished as it appears, that the things we believe we have fully understood are merely the things we have fully exhausted the tools to interrogate, and that every generation of inquiry, armed with instruments its predecessors could not have imagined, will find something in the old evidence that the old methods could never see. The Dead Sea Scrolls have been studied for nearly 80 years. Generations of brilliant scholars have devoted their careers to these fragments, and yet it took a swab of degraded animal DNA, a computer algorithm, and a team of molecular biologists to reveal a truth that all of that scholarship had missed. Not because the scholars were not brilliant enough, but because the question they were asking required a language that history had not yet learned to speak. That language is now available to us. And if what it has already told us about 900 ancient manuscripts is this staggering, one can only wonder what it will tell us next. What other objects, what other artifacts, what other seemingly exhausted archives are quietly holding biological secrets in their fibers, waiting for a question no one has thought to ask yet. The shepherds who first stumbled into those caves in 1947 could not have known what they had found. The scholars who spent the following decades piecing those fragments together could not have imagined that their work would one day be continued not in reading rooms and archives, but in genomics, laboratories. And the scribes who pressed those words into skin 2,000 years ago could not have foreseen that the very bodies of the animals they slaughtered to make their books would one day become a kind of testimony in their own right. But that is exactly what happened. The skin remembered. The DNA endured, and in the silence between the letters, in the molecular structure of a hide stitched together from 2 distant herds, a civilization that thought it had been erased left one final, extraordinary message. We were here, we were many, we were complicated, and we did not go quietly into the dark.